My name is Gonzalez, and my project is Insights into Genetic Connectivity of Great Sugarfish, or Balistes capriscus, within in artificial reefs of the Gulf of Mexico. I do want to apologize for giving everyone the bait and switch. What I had submitted to the EPP forum abstract was something a little different. It was going to incorporate stomach DNA. So if those of you came for that aspect of it, I do apologize. Originally what had happened was my project was essentially this within the northwestern Gulf of Mexico. And shortly after doing my synopsis and whatnot, when I was doing research for my thesis proposal, it ended up being that my project had been taken up at that point by a larger project that encompassed the entire region of the Gulf of Mexico within the US. So therefore, no longer needed my research in that area. So in order to make it viable, we tried to incorporate another aspect of it, which is how we came up with that abstract from before. At some point, after conversing with my professor, we ended up being able to acquire samples from Mexico, which was an area that was not able to be studied or incorporated into the previous study, therefore making this a viable study that would be beneficial in understanding the whole effect of the movement in the Gulf of Mexico. So that's how we got to this point, and I will continue from there. So a little background: artificial reefs have been deployed as a method to help mitigate habitat loss. Not just mitigate habitat loss, but provide new habitats. You know, there's plenty of areas within the Gulf of Mexico that do not have any type of hard substrate. So artificial reefs can be considered anything man-made that's going to be put into the ocean that's going to help the marine community down there. It's going to help them thrive. It's going to provide basically an area for them to thrive. And Balistes capriscus are not considered a relevant commercial fishery species in the northwestern Gulf of Mexico, but with increasing fishing pressures on other fish, such as the red snapper, they are more constantly targeted, and that's essentially the relevance of them. So a little bit of their life history. There are wide-reaching areas across the globe, which we'll go into a habitat map in a moment. They are considered a single stock within the northwestern Gulf of Mexico, which is why we're doing this research. And they are associated with hard benthic areas such as reefs, both natural and artificial. And as you can see from their teeth, they have very ferocious looking teeth, which doesn't necessarily mean that they're carnivores or anything like that. More so, it just allows them to be opportunistic and eat whatever is available, including things such as sand dollars or urchins and whatnot with those hard exoskeletons. So going back to the habitats, this is a habitat map. As you can see, it's far reaching. And this is essentially going to be part of the reason why we're studying, I guess, the connectivity, because as you can see, it is far reaching, and that plays a part into the gyres. So gyres are going to help distribute their larvae. Oh, sorry, I feel like I'm getting ahead of myself. Okay, so their larvae are going to hatch, which we'll go into a moment. But actually, let me just show you a distribution map of the gyres. So we have the North Atlantic gyre and the South Atlantic gyre, which are going to contribute to their distribution. As you can see, they follow similar patterns. These areas that the gyres touch are areas that we also see within their distribution. A little more information about, I guess, Balistes capriscus are that they're managed by Reef Fish of the Gulf of Mexico Fisheries Management Plan set in place by NOAA. This is they have minimum size limits, bag limits, seasonal closures in place for commercial and recreational catches, and that's just to protect these fish. While they're not considered overfished, they are still rebuilding to target levels. And for that reason, that is why we have limits in place. Reproduction. So they spawn in the Gulf of Mexico, and this occurs during May through August and peaks in June and July, and they form harems for reproduction. And if you're familiar with a harem, that's essentially just one male to several females within the Gulf of Mexico in our area that will originally started. They do maintain a 1-to-1 sex ratio, so one female for every one male. All that means is that there's going to be some males without partners, there's going to be some males with multiple partners, and the larger the fish, the more viable. So the larger fish will have more, I guess, female representatives. Once hatched, larvae travel to sargassum and use them as a nursery ground. This can last for several months, 4 to 7, and after which they'll relocate to benthic communities, which they'll spend the duration of their life. I don't mean to keep going back and forth, but if you're familiar with sargassum, there's a sargasso sea which is located in the North Atlantic gyre. And so if they're located in the gyre, in the sargassum, that sargassum is going to be carried around with these gyres, and that's going to attribute to their high population and as well as their single or their status as a single stock. If it has high level of dispersal, there's several months in there, then they can reach pretty far areas that would be, I guess, unable to be reached if they had like another means of transportation. Afterwards, as we mentioned, like in their adult life, they are pretty sedentary. They'll relocate to a bottom hard substrate, pretty much spend the duration of their life. They're pretty territorial. They're not going to necessarily want to move anywhere else. And if you approach them, which I have in a diving trips, they will definitely come at you, try biting you, or even try poking you with their spine. Leave it on this side. So study area, because the Gulf of Mexico lacks naturally occurring hard substrates, such as naturally occurring reefs. Artificial reefs will be the connecting points of the distribution of these reef related species. I do want to point out that whenever this project started, and we will try to keep it consistent, which would be finding artificial reefs within our other areas, but I'll get into that in a moment. But two of our reefs will be off the coast of South Padre. These will be the Texas Clipper and the RGB Reef. One will be near shore, one will be offshore, and then we'll also be getting samples from Mexico off the coast of Quintana Roo and off the coast of Veracruz. So if you remember the gyres and the circulation pattern, we can see that we'd start off in Quintana Roo, potentially, maybe Veracruz, and then we'll go upwards towards the areas that we're located, which would be off the coast of South Padre, which is the two stars located up there, representing our RGB reef and our Texas Clipper. So with that being said, we are not exactly sure if the samples we'll be collecting from Mexico will be located on an artificial reef. So for that reason, this project may be changing to reefs in general instead of specifically artificial reefs. So this is a picture of the Gulf of Mexico currents, and this is going to be based on seasonality. So you have January through March for the first one. You don't see too much water movement. You have April through June. You start seeing it increasing. July through September, it's very active, and then you start seeing it slow down from October to December. So this is important when you think about their when they reproduce, which if we remember from the previous slide, is between June and July, typically when they have the highest spawning. So we'll take a further look at the July. And just to keep in mind, they do travel for about 4 to 7 months. So that's a long period to be traveling. And then based on the time period or however long that they stay in there, that's going to determine how far they're going to move. So if, let's say, a weather event happens, we have a hurricane, then that's going to disrupt this pattern. So it's not going to be consistent, but it will, I guess, basically give a good indicator of the areas that these, the sargassum with those larvae and juvenile fish will be reaching. So ocean plane, as I guess insinuated, ocean currents play a large role in the distribution of the capriscus, and understanding how they're distributed can lead to predictions in their genetic activity. So the rationale for this would be that this research will help fill knowledge gaps. As mentioned, research has started to get done into this, so we'll be adding to that, and improving our understanding of gene flow within the lower Gulf of Mexico is important when assessing their vulnerability. So this goes back to understanding, I guess, where they're coming from and where they lead to. If we understand how Balistes capriscus are being affected in certain areas, let's say we are getting our supply from Mexico, then if we see a decrease in those populations, well, then how will that affect us? Or more so, like if we see a decrease in our populations, we experience overfishing here, how will that affect other areas? There might even be a case where, if you remember from this image, there's areas that look like they don't have so much flow. Does that mean that those area populations are going to experience less connectivity from other areas as they start to differentiate within the fact that they're, I guess, multiplying within that specific area? So I guess with those questions, with this research, we'll be answering those questions. So our hypothesis at the moment is that genetic markers will indicate no significant difference between populations and capriscus. That's based on the research that's been presented so far, as well as previous research into the study. Goal and significance, we will be adding to current studies of the capriscus population structure within the Gulf of Mexico. It could contribute to further understanding of the ecosystem processes, impact of the capriscus genetic connectivity, and provide information useful for future management. So going into our sample design, fish will be caught using the hook line approach and collapsible fish traps. We've already been out to the field, and using these methods, we were able to catch high numbers. Therefore, we will be using this moving forward. Currently, our target is about 30 fish per site. And in total, we should have about 120, we will have 120. And upon capture, they will be sorted in an ice slurry and processed until arrival to shore, which in that point, we will extract a 1 by 1 centimeter muscle tissue sample and store that in alcohol. The alcohol will be replaced, and we will keep these in Eppendorf tubes at -20 degrees Celsius until we extract the DNA. So as mentioned about previous work, Salient was the one to do the large study. He created these 17 microsatellites, which are depicted in the grayscale. We ended up finding another paper by Sorensen, which would be located in blue scale, where she detected 8 more microsatellites that would be useful for detecting that divergence in genetic material. Just a larger size, that way you can get a better idea of what those were. So this is Salient. And right here would be Sorensen. So with those microsatellites, we'd be using SAMNOVA to distinguish potential divergence between populations. And then we'll also be using the mitochondrial DNA in order to assess the homogeneity within localities. So in one area, we'll be seeing how similar they are with each other. And then we'll be using the microsatellites to determine how far apart they are from other localities. And with that, I would like to thank NOAA and CCME for this opportunity. I'd also like to thank my professor, Dr. Sintra, for his encouragement, as well as getting us through all the hurdles that have come about. With that being said, this is very much still in the preliminary stages. So if there's anybody that's familiar with, I guess, projects similar to this or has any type of input that they'd like to contribute, please let me know, reach out through email or through conversation, and I am more than willing to take any type of, helpful feedback that might be beneficial.
Are there any questions?
Oh, sorry. Ready?
So I'm a physics circulation kind of guy, so
Okay.
I look at bioconnectivity from currents.
Okay.
But one of the questions I have is just probably, I'm an uninformed type of person. Will you be able, with your type of analysis, to just be able to say that they're connected, or will you be able to determine what would be a source population for maybe a downstream population?
That's a good question. Honestly, I'm not too sure. I'd have to ask, since this is so preliminary, I still need to get with my my team just to see exactly. We have a fish geneticist as well as a invertebrate geneticist, and I think they'd be able to, I guess, help with that type of question. At the moment, I'm not too familiar with that, but that is a good question to ask. Thank you.
We have time for a couple more.
Oh,
Yeah, I'm curious if you think the impact of hurricanes would also contribute to how they're circulated,
Most
or if that's
definitely.
encapsulated in the circulation.
No, so that was mentioned, weather events, hurricanes would disrupt the flow. I guess especially during that time period, that's the hotter months, we're gonna experience hurricanes and such, weather events that would disrupt the normal circulation pattern. So most definitely, you would see if maybe it's gonna hinder them from moving to other areas and keep them in that locality, or maybe they'll get out in time and it's gonna push them further away. So I mean, that's definitely something to consider.
Maybe one more question?
Great presentation, Jacob.
Thank you.
I'm just curious to the passive transport mechanism associated with these triggerfish and how likelihood or what's the overall likelihood of those larvae reaching their spawning grounds basically, and how do you have an idea of how likely they'll be able to passively transport to their native fertilization point? Do you understand all my question?
A little bit. So you're asking how, what's the likelihood of them traveling back to their original area?
Right. Because I'm thinking since they're passively transporting, I'm assuming that these larvae are just a product of their environment, so they could have picked up the currents and other hydrodynamic forces. So I'm assuming since those contribute to the spatial distribution, that those larvae might be distributed differently and contribute to like stock variability in their genetics. So I'm just curious to see like how what's the likelihood of those larvae reaching their native parent grounds.
I feel like the likelihood of them reaching their native ground would be highly unlikely. I feel like if they are able to stay within that area, like if there's a little circulation within that area, so they're not like traveling, then they would just stay within that locality. But if they're going to get caught in the current and get taken away, I don't anticipate that they'd likely return to the same area. But that's just my, interpretation. I have no work to back that up. That would just be my opinion.
Definitely. Thank you.
Thank you.
Thank you.